Right. This is video number 2 in the group on Introduction to Microbiology for surgical technology students. And we're going to start talking about the different types of microorganisms. Here we're going to start out with bacteria, as I mentioned earlier in the previous video. This is the largest section that we will discuss. And so it's going to be video by itself, and then we'll do the other microorganisms separate. All right, first we want to talk about the morphology or the shape of bacterium. Um, these are single-cell prokaryotes, and so remember prokaryotes just means that they're not as sophisticated, not as complex of cells, and they have their DNA is not enclosed in a membrane. Of these, they do tend to form in different groups, chains, clusters. They can be in groups of 2. There's different arrangements. And so when we're looking at the shapes, we're going to look at the shape or the morphology combined with groupings to get a description of the bacteria. And so we've got 3 main shapes: coccus, bacillus, and spirilla. And luckily these aren't too hard to keep up with, as as the names. The coccus is a spherical, so like a ball shaped. These come in chains and clusters for the most part, but you'll see some that are individual. You'll see some that are diplopia. That's going to be a 2 coccus connections. So we'll see these in some different arrangements, pretty common. Then we've got our rods or bacillus, and so these are rod-shaped, and then spiral-shaped or spirillum. Now most of these will multiply by binary fission, so you only have to have 1 bacterium in order to make as many as you want. So you don't have to have 2 that get together to create a third one. All you need is 1, and that 1 can divide to form 2, and then that 2 can divide to form 4, and so you get a really rapid multiplication here. They can move by using flagella, and they can get around pretty fast with those too. So they are in a small world, but if you're moving fast in a small world, you may not be moving far, but you are moving fast. So a lot of them do have flagella. All right, so here's some pictures showing some of these different types, and these are going to be like what you look at under a microscope. So if you're doing a basic staining, and we're going to talk in a minute about gram staining, which is our most common form of staining the bacteria so that we can see them. And so we'll stain them so we've got a color, and then we can see the shape. And this is not the strongest magnification by any means, but under standard microscope, you will be able to see the shape and the arrangement. And so this one up here is a diplococcus, so the 2s. And you'll notice that there's some that are by themselves, and there are some chains that happen to get together in there. But the majority of these are running around in pairs. I just marked right through that one, but you get the idea. These are staphylococcus, so this is that grape-like cluster of the spheres. And here again, you'll notice there's some that are broken loose, and there's some that are not connected to many, but the most prominent groupings of these are clusters. And they're spherical, so that's coccus. Clusters are staph, so staphylococcus. And then this is streptococcus. So in chains, this is a little bit stronger magnification, so you can see those a little bit better. But the spheres and in chains. So strep for chains, and coccus for the spheres, so streptococcus. Oops, I'm sorry, I went past one. And so this is a little bit bigger, a little bit better magnification with a standard microscope here on the left. And so you can see the Staphylococcus aureus here, which is one form of Staphylococcus. And they are in those clusters and spheres. And then this on the right is electron microscope version, where you can see it much more in detail. And so you can actually see how they are spherical. These have also been stained and with gram stain, so these are gram positive. Staphylococcus is a gram positive coccus. And you can see those purple spheres in a grape-like cluster. Bacillus, rod-shaped, and so this under standard microscope it's a little bit harder to see, but this is a gram-negative bacillus, and those are rod-shaped. And then electron microscope version, where you can see much more in detail, and you can even see the flagella on these, so much closer detail there. And so this is E. coli, one of the ones we'll talk about later. This normally resides in your intestine. And so these are bacilla, gram positive, gram negative bacilla, and that is E. coli. And then I guess one of the coolest ones, the spirilla, spiral shaped, and so definitely able to detect that shape. Now, in order to get these colors, we're going to use gram stain or use staining techniques. You can do just a simple stain that stains all the bacteria one color. But the gram stain allows us to do a differential stain where we can tell by the cell structures or the cell membrane whether or not it will take the pink dye or the purple dye, thus being gram positive or gram negative. Now we have other type of differential stainings. Acid-fast stains are more for bacterium that have a waxy coat around them that will need to be dissolved a little bit before you can actually get the stain to attach. And then we've got some that can do just the structures of the cells. And then we've got immunofluorescent or fluorescent dye stains that can also be used. But the most common one being the gram stain. And this is something we're going to do a lot. We're not going to do a lot, but it's done a lot in healthcare. So we will send, if we culture an infection, and we want to get some idea of what it is. We may kind of know, but we want to make sure that the patient's getting the best antibiotics to begin with. Well, some antibiotics work better with gram-positive bacteria. Some work better with gram-negative. And if we can find out if it's gram-positive, gram-negative, and then the shape and arrangement of that bacteria, then we can come a lot closer to getting an idea of what the bacterium is and what antibiotics would work for it. And so this is called a gram stain because it's named after Dr. Graham. And so no fanciness to the name. It's just named after the person that developed it. And so this is a process where you take a microscope slide, we take some of the infectious agent that's diluted, placed on the microscope slide, and then spread across it. The microscope slide is then heat fixed, so that bacterium is fixed to it, dried to it. And then we'll go through the different steps of dyeing those bacteria. So to begin with, the whole microscope slide is flooded with crystal violet, and as you get the idea they're violet, it is a purple color that stains all the cells purple. Then we're going to add iodine. Now the iodine is a mordant that helps to seal that purple color in those gram-positive bacteria. The slide then is rinsed with alcohol. The gram-positive cells keep the purple color. The gram-negative cells become colorless. And then another stain is added, saffron, which gives the pink color. So if it's gram-negative and we've made it clear after rinsing it with alcohol, then that pink stain will attach coloring your gram-negatives pink. And so here's a picture of two different bacterium together in the same slide, where you can see the gram-positive purple and the gram-negative that is pink. And then this goes through, and I think the picture kind of gives you a little bit better visual of the technique, because it kind of shows you an example of what would be happening with the two microscope slides here. So as they are first fixed to the slide, they're both transparent. And then the purple stain, crystal violet, is added, both are purple. And then it is coated with the iodine, which fixes it. The mordant, the iodine, meaning that the mordant, meaning that fixes that color in the gram-positive. And then it can be rinsed with the ethanol, with alcohol, and the gram-negative then become clear, but the gram-positive stay purple. Then when we add the saffron, that pink color is not going to change the color of the purple, as it is already stained, but it will change the color of the colorless ones, the gram-negative, and make them purple. And so then that allows us to be able to better identify. So we do the gram stain, and then you can look at, is it gram-positive, gram-negative, and then what type of shape and arrangement, and it gives us a direction to go on the antibiotics. Now, so this is something that can be done pretty quick. So when we send a culture, and when you're in the scrub row, if you are doing a procedure and you know there's going to be infection, then you're probably going to need a culturette on the field. So this is a little plastic tube that has two swabs in it, 1 to 2 swabs, according to which one it is, if it's a big one or a small one. And when you come upon the infection, you hand that swabs to the doctor, they swab the infection, hand it back to you. You put it in that tube, and they have different ways. Some of them have a glass ampule that you break, and others just have a second tube that you put them in that will have some liquid in it that will keep the bacterium alive until you get that to the lab, to microbiology. And so that's done usually early in the procedure, when you first encounter the infection. And that's handed off to your circulator, the nurse in the room, and that should be processed and sent immediately to the lab. When that's done, it should take about an hour to get the results of the gram stain back. And so you may still be, during the procedure, still working. And if it's been a while, the doctor asks, can you check and see if the results of the gram stain are back? And so, like I said, about an hour, you should be able to get those back. But one of the important parts is immediately handing off that culture as quick as you can, and the circulator RN, getting that processed and sent to the lab for a stat gram stain. The other reason you don't want to hang on to that culture tube is that stuff continues to grow in that tube. And so we really want to get a good representation of what was in the patient. And so we want to send it on so that they can go ahead and do that gram stain. Now, they will typically also order a culture and sensitivity at the same time, and so we're going to cover that here in just a minute. But the acid-fast stain I mentioned earlier, very much the same, similar to a gram stain, except that you have to use additional steps of additional chemicals in order to remove the waxy lipids that are surrounding the bacteria that will keep the dyes from actually connecting to those. So very similar if it's indicated. Sometimes the doctor will order, multiple gram stain, acid fast, both, or the acid fast if indicated if the gram stain doesn't show. And so, anyway, so very similar, gives you the same information. All right, motility, movement. Like I said earlier, some of these have flagella. E. coli is one of those. It can move at 20 body lengths per second. Like I said, really fast. Doesn't get very far, but really fast. And there's other things that's going to help the bacteria to move. So a lot of times they are attracted to certain chemicals and remove towards or away from that, but they can also move towards or away light, air, or magnetic fields. So all those things can cause movement of the bacteria. And then some will have pili instead of flagella, which are just shorter, thinner hair-like structures that help with the movement. All right, colonization. So as these spread from that initial inoculum, it will grow on an agar plate in these little round cells of colonies. Now, if you put one in the middle of a spot, then that colony will grow out from that, and you'll have just a single colony. So it's not easy if you just smear a bunch of it on there. You're not going to you're going to have the colonies overlapping. So we're going to talk about that here in just a second. But they do tend to come out from create a ring of colony of the bacteria. And one of the ways that we get down to isolating just the one bacterium and getting little individual colonies is using the streak plate method. And so this is simply performed, and here's a diagram, and here's a plate that's been already done and incubated. But you start out with some of the microorganism on a sterilized loop and start by streaking that back and forth through one portion of the agar plate. Then you re-sterilize the loop, and then you just track through the edge of that. So you see how it's just kind of dipped into the edge and then back and forth. And then that is continued on around the plate. And each time sterilizing. So you see how this continues until you get to that last little bit, and sterilized loops taken into the edge of that where it's already been diluted, and then kind of out through the middle. And so then that allows you, as you can see here, there's a good example of an individual colony that is separated. So that'd be one colony, all of which is one particular bacterium. And so you can look at that and see which one, or identify the shape and arrangement of that bacterium and get a little bit more information about it. Whoops, went too far. So culture and sensitivity. Then we can streak a plate totally covered in that isolated bacterium, and then add antibiotic circles. And what we're looking for here, we say the term culture and sensitivity. Culture means that we culture the plate. We've covered an agar plate with the bacterium and let it grow. Sensitivity means which ones actually affect that bacterium. So is the bacterium sensitive to something? Is it affected by that particular antibiotic? And so this can be done by placing antibiotic disc onto that plate. And as you see, these areas of halo around these disc are various sizes of areas here. And those are the antibiotics to which this bacterium is sensitive to. So that means that that antibiotic will work against that bacterium. And you see the ones with the bigger rings seem to be working better. So that's the ones with the greatest sensitivity. So that would be your preferred antibiotics there, the ones with the biggest rings around them. And so that's giving you that area of no growth. So that halo area is an area of no growth. Now this is how penicillin was discovered, in that it was in a lab environment. So I guess I should ask, who discovered penicillin? I'll give you a minute, look that one up. All right, Sir Alexander Fleming, in the late 1920s, was working in a lab and had some cultured plates, and one was sitting, or some that were sitting close to an open window, had some mold had gotten onto it, had gotten contaminated with some mold, and the mold started to grow on the plate. But the thing that didn't grow around that, so where the mold was, there was a halo around the mold. And so he, from that, realized that there was something in that mold that was killing the bacteria. And thus our first antibiotic, penicillin, was then came from that discovery, and was extracted from that type of mold. Now, another way we can do this is rapid antibiotic susceptibility testing. And so this is just another way to do a sensitivity. And in this allows a little bit faster looking at many different antibiotics to see which one of them which ones will work. And with these, you have these trays, and these are done in the lab, of course, in micro. And when I say the lab, there's different areas of the lab, and micro is one of the big ones. That's the ones that does the all the cultures and gram stains and etc. We also have chemistry in the lab. We've got the blood bank. We've got pathology. So a good bit going on. But in each of these little vials in this, these little, sorry, losing the word that I'm looking for. Wells, they're little bitty wells. So anyway, they're just these little cup-like things that, and in those you'll have the different antibiotics and a medium, a growth medium. And so to those, they can add just a small amount of the agent that we're testing, the infectious agent, into each one of those, and then they allow that to incubate and watch for a color change. And the ones that have growth will have a color change. The ones that do not then would be the ones that were effective against that microorganism. Now, speaking of growth, some things that affect growth. Now, think about this, and the human body on the inside is an excellent place for growth of microorganisms. So, especially when you get, something such as Staph aureus off the skin, and that gets down in that subcutaneous layer of tissue. So subcutaneous is the fatty layer. So you got skin, and then you got the subcutaneous. Then you got muscle, fascia, peritoneum. You're then inside the abdominal cavity. So you're making an incision into the abdominal cavity. You make the incision through the skin. Let's say that you get some of that staph off the skin. It gets into that fatty layer. At the end of the case, we always irrigate the belly. Then as we start to close, once we close the muscle and fascia, we irrigate again that subcutaneous layer, because we can also get bacterium from the bowel when we're doing like a colon resection or a small bowel resection. So there's a lot of bacteria in those, and we use techniques to try to reduce how much of that we cross-contaminate. So we'll use bowel technique, in order to reduce that. We will irrigate in order to reduce that. But there's still that risk of getting a bacterium transferred back up in that subcutaneous layer. Well, fat is not very vascular. So one of the best things about fighting infection is good blood flow. Good blood flow brings antibiotics, it brings oxygen to the area, helps it heal. And so tissues with good blood flow, much more resilient to infection. Tissues with poor blood flow, not very resilient. And so the subcutaneous layer, not very resilient. So anyway, we pick up some of the Staph aureus, we get in that layer. Well, the body temperature, nice temperature on the inside, nicely controlled, nice moist environment. It has the perfect amount of oxygen. It's got the perfect amount of everything to then start growing. So all those things can affect the growth of a microorganism. Oxygen, pH, water, and temperature. Now, I like this chart. You've got to understand that, and we mentioned earlier, bacterium divide by binary fission. So you only have to have one. Now, this depends a lot on variables, and it also is highly unlikely that we're going to get just one bacterium in some area, such as the subcutaneous layer, and the body not be able to fight off that one. But, that one can potentially, and the thing is, if we get multiple, then that really does increase the odds of this happening. But if we have one that divides, and they divide every 20 minutes, bacterium do. And so if you start with one, at 20 minutes you got 2, 40 minutes you've got 4, at an hour you've got 8. It doesn't take long. In 7 hours, you've got over 2 million bacterium. And so, as you learn in lab and when, in class when we talk about surgical asepsis and reducing the risk of infection, and we teach you these things in lab, we're doing it for a reason in that, you never know when, a breach of asepsis could create this, and you can't have breaches of asepsis as it runs a risk of getting this started in a patient. And then many of our patients already have weakened, immune systems and don't feel good, so their body's down a little bit and it's not as capable of fighting infection. So, it's one of those things that, infection doesn't take much to get, something really started. All right, we do like to classify our bacteria as aerobic and anaerobic. And there's different levels of these, and then there's other gases also. We'll go through several more definitions here, but the biggest two of them you definitely have to know, and then we'll go through the other ones that are good to also be familiar with. But you definitely always need to know that the aerobic need oxygen, anaerobic will not grow in the presence of oxygen. So they prefer not to be or do not need to be around oxygen. So now this comes in different levels though oops, going a little too far, sorry. All right, here we go. All right, and so there's a few different ones here. So microaerophils require low oxygen, less than what's in your room air, but do need a little bit of oxygen. And then we've got obligate anaerobes, meaning that they will not grow with any amount of oxygen. So they are obligated to be an anaerobic. So they do not have a choice. They're totally anaerobic no matter what. And then you've got the facultative anaerobes. And so facultative, they can kind of adjust. And so they don't really like much oxygen, but they can survive in some oxygen or no oxygen environment. So they prefer not to have oxygen. They're anaerobic, but they can adjust. So they're facultative. They can adjust. And then capnophils, anytime you see this term capnode, that is CO2 or carbon dioxide. So capnophils grow best in high concentrations of carbon dioxide. All right, one thing that is important when we start looking at sterilization is endospores. With bacteria, some bacteria, and for the most part, these are bacillus, the rod-shaped bacteria, have a way of creating a spore when they'll subdivide and then place that DNA material into a small, separate, membrane reinforced, highly resilient spore. And so you can see the bacillus here creating endospores. You can see the little darker dots. Those are the endospores that have been created by these bacillus. Now, why do they do this? Well, they're getting into a situation where their environment's drying up too much, and they're like, ooh, we're not going to be able to survive in this dry environment. Okay? And so they'll start to then create or produce those endospores. Then the endospores can survive the extreme conditions for any amount of time. It can these can survive in the frozen tundra. They can survive in really high saltwater areas. They can survive many places. And then when placed in an ideal condition, they go back to growing. And so they'll come back to life. So all they need is that moisture and the right temperature, right oxygen, and they'll go back to growing. So endospores are pretty dangerous because they are in that dormant phase while they're a spore, and then they go back into a living vegetative bacterial growing, reproducing phase then when they get back in a good environment. Well, the thing about this is since endospores are made to be resilient to the tougher, more extreme environments, they are the hardest to kill by sterilization. So this is what John Tyndall realized when he realized that bacteria can be in two different states, is he was seeing or recognizing these endospores. So when they boiled a hay infusion and they didn't boil it for long enough, they would kill the living bacteria that were in that hay infusion, but they would not kill or destroy the spores. And so then they would boil it, kill the living bacteria, but then since it's a broth, the spores would come back to life and become living vegetative bacteria and start to reproduce and grow. And so that's where there was the variance in results with the different people that tested these theories, is that if they used a broth that had a lot of bacillus in it, and it was one that formed a spore, then they wouldn't destroy the spores if they didn't boil it for an extended period of time. And so this is also true with sterilization. It's that sterilization has to go long enough to kill even a spore. And so when we talk about sterilization in chapter 7, the only way we can truly know that something is sterile is if we can kill spores with the sterilizer. And so something is not sterile unless it kills all microorganisms, including spores. So when we get to chapter 7, we'll talk about high-level disinfection. That kills all microorganisms except spores. So spores are not destroyed by high-level disinfectants. That's okay if it's such as, for using an endoscope, such as for colonoscopy, because the inside of your GI tract, your mucous membranes are resistant to spores, so then it's not going to cause an infection. However, if you get spores in other areas of the body that they're not supposed to be, then they can go back, or they will start to grow. They'll become back a living bacteria. And so we have to use sterile stuff, instruments, supplies, anytime we're operating in areas of the body that would be other than a mucous membrane area, that's such as looking into the mouth, the throat, looking into the lungs, or into the colon. All right. So in order to do that, we've got to kill all microorganisms and the spores. So we test our sterilizers on a regular basis. Really should be done on a daily basis, but according to your facility and how often they use the sterilizers, that can change. But we actually take what we call a biological test, and this is a little ampule that has a special bacillus. The Stereobacillus stereomophilus is a bacillus that would then take the spore form of that bacillus, and it's placed in this little tube. These come manufactured from us. 3M makes one of the most popular ones. It's called an A test. And we'll put that in the autoclave along with whatever we're sterilizing, or we'll put one in the autoclave once a day just to make sure that the autoclave is working and that it's sterilizing everything inside. And so that is run through the autoclave run. And then it is taken, it's got a little ampule, glass ampule on the inside of growth medium. There's a little crusher on the machine that we read these in. It's placed in an incubator well, and then we'll come back and check it for growth of those spores. Because if it did not kill the spores, once we apply that growth medium to it and place it into an incubator, those spores will become live and active and start to grow. And so the A-test machine gives us a readout of positive or negative, but you can also do these biologicals with even test tubes of a growth medium with the spore in there that has come through the autoclave run, and then we watch for a color change or for a cloudiness of it according to what type that you're using. And so if it did not kill all the spores and it starts to grow, then that sterilizer is no longer sterilizing effectively, and it needs to be worked on and corrected. And so if we're running instruments that we really want to assure that that particular run, such as if it's implants, we'll run a biological every time we run implants. Some hospitals will run a biological with every load of instruments that they run. Others will run one every day, but then those instruments are tracked. So if they get a bad biological, they pull all the instruments from that run from that time back to the last good biological. And so anyway, this is a very important part of our definition of sterility, the biological test, which are ultimate tests that the autoclaves are working. So that goes back to endospores. All right, nutrition, things that bacteria need to live: carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, magnesium, calcium, and iron. They also carbon in the form of glucose and carbon dioxide, and then some will get energy from light and chemicals. And then pathogenicity and occurrence. Of course, these are wide range. from extreme systemic infections that can cause organ shutdown and death, to those that live with us and on us and in us every single day. So Staph aureus, Staph epidermidis, lives on our skin every day just as happy as it can be. We need it on our skin, good for our skin. In our intestines, we have E. coli that are natural to our intestines, that are important that they're there. We've got to have bacteria in our intestines to help break down food. Now, you can get an E. coli infection from the wrong E. coli. We'll save that one for a little bit later when we talk about the different microorganisms that cause infections. All right, so we can find these in extreme places. So even in the Dead Sea, we get this super high salt concentration. There are bacteria that will live there. So wide range, lots of bacteria, a lot more different bacteria out there. I tell you what, they're everywhere. All right, so this ends video 2, and then we're going to start looking at the other microorganisms in video 3.